So as we take a look at cell signaling, there are a variety of different responses that can be elicited from one cell signal. What's that cell supposed to do once it receives a specific signal? Remember, there's three different parts to cell signaling: reception, transduction, and response. We're going to focus on reception in this brief video. So in reception, we basically have to capture a signal molecule. Signal molecules attach to proteins that are embedded in the plasma membrane, or at least can be embedded in the plasma membrane. The first type that we're going to take a look at is called the G protein coupled receptor. It is a transmembrane receptor, and as you can see here, we have the one on the left doesn't have the ligand as yet. The one on the right does. So let's first take a look at the left. Here is the outer portion of so this is the extracellular fluid out here. Okay. This is the intracellular area. This is inside. Okay, so here's your plasma membrane. So this particular protein twists back and forth several times through the plasma membrane. When the ligand is present, that's going to change the conformation of that protein, okay, causing the end of this protein to change right here. And so by receiving a ligand that never enters the cell, we change the composition, the conformation of this protein, releasing things into the cell that begin that cascade of transduction, sending the message throughout the cell. Next we have a chance to figure out how to clear the screen. There. Okay. Now, next are our RTKs. RTK is short for receptor tyrosine kinase. These things have a very special way about them. Let's take a look. The best way here is a picture. So first, we have two inactive monomers here. When the ligand shows up, okay, when the ligand is in that extracellular fluid, so let's establish where we're at here. This is this is extracellular fluid. This is inside the cell. Okay, this is our plasma membrane. Okay. The ligand binds to both of these monomers, bringing them together, causing a dimer to be formed. Okay, two things put together. Di means two in chemistry. Okay, that is going to cause the conformational change in these proteins that allow them then to behave as docking sites for these phosphate groups. Okay. And the phosphate groups are going to be significant in the ATP-ADP cycle here and causing things to happen further inside the cell. That is the RTK receptor. Okay. So again, the RTK receptor is significant because it brings those two monomers together, forming that dimer, the dimerization, okay? And that causes all sorts of pathways to open up because it triggers that phosphorylation action within the cell.